Hello everyone, I am Aishita from Ixpoe. The topic which we are going to study today is photosynthesis, which is a continuation of our previously uploaded videos. The process of photosynthesis, carbon fixation and reduction. The assimilation of carbon into organic compounds is the result of a complex series of enzymatically regulated chemical reactions, the dark reaction. The term is something of a misnomer for these reaction can take place in either light or darkness. Furthermore, some of the enzymes evolved in the so-called dark reactions become inactive in prolonged darkness. However, they are activated when the leaves that contain them are exposed to light. Elucidation of the carbon pathway. Radioactive isotopes of carbon and phosphorus have been valuable in identifying the intermediate compound formed during carbon assimilation. A photosynthesizing plant does not strongly discriminate between the most abundant natural carbon isotopes and carbon. During photosynthesis, the presence of carbon dioxide, the compound formed become labeled with the radioisotope. During very short exposure, only the first intermediate in the carbon fixing pathway become labeled. Early investigations showed that some radioactive products were formed even when the light was turned off and the carbon dioxide was added just afterwards in the dark, confirming the nature of the carbon fixation as a dark reaction. American biochemist Melvin Kelvin, a Nobel Prize recipient for his work on the carbon reduction cycle, allowed green plants to photosynthesis in the presence of radioactive carbon dioxide for a few seconds under various experimental conditions. Products that become labeled with radioactive carbon during Kelvin's experiment included a 3 carbon compound called 3-phosphoglycerate, sugar phosphate, amino acid sucrose, and carboxylic acid. When photosynthesis was stopped after 2 seconds, the principal radioactive product was PGA, which therefore was identified as the first stable compound formed during carbon dioxide fixation in green plant. PGA is a 3 carbon compound and the mode of photosynthesis is thus referred to as C3. In the other known pathways C4 and CAM, the C3 pathway follows the fixation of carbon into oxaloacetate, a 4 carbon acid, and its reduction to malate. PG is formed from 2-carboxy 3-keto D-arabinitol 1,5 biphosphate, which is a highly unstable 6 carbon compound formed from the carboxylation of ribulose 1,5-bisphosphate, a 5 carbon compound. Further studies with C14 has or as or well as with inorganic phosphate labeled with 32P lead to the mapping of the carbon fixation and reduction pathway called reductive pentose phosphate RPP cycle or the Kelvin-Benson cycle. An additional pathway of carbon transport in certain plant was later discovered in the laboratories. All the steps in these pathways can carried out in the laboratory by isolated enzymes in the dark. Several steps required the ATP or NADPH generated by light reaction. In addition, some of the enzymes are fully active only when conditions stimulate those in green cells exposed to light. In living plants, these enzymes are active during photosynthesis but not in the dark. The Kelvin-Benson cycle. The Kelvin-Benson cycle in which carbon is fixed, reduced, and utilized involves the formation of intermediate sugar phosphate in a cyclic sequence. One complete cycle incorporates 3 molecules of carbon dioxide and produce 1 molecule of 3 carbon compound glyceraldehyde 3-phosphate gal3p. This 3 carbon sugar phosphate usually is either exported from the chloroplast or converted to starch inside the chloroplast. ATP and NADPH formed during the light reaction are utilized for the key steps in this pathway and provide the energy and reducing equivalents, that is, electrons, to drive the sequence in the directions showed. For each molecule of carbon dioxide that is fixed, 2 molecules of NADPH and 3 molecules of ATP from the light reaction are required. The overall reaction can be represented and follow. 9 ATP plus 6 NADPH plus 3 CO2 gives rise to Gal 3P plus 6 NADP plus plus 9 ATP plus 8 phosphorus. The cycle is composed of four stages: carboxylation, reduction, isomerization, condensation, dismutation, and phosphorylation. Carboxylation, the initial incorporation of carbon dioxide, which is catalyzed by enzyme ribulose 1,5 bisphosphate carboxylates, which is Rubisco, proceed by the addition of carbon dioxide into 5 carbon compound ribulose 1,5 bisphosphate RUBP and the splitting of the resulting 6 carbon compound into two molecules of PGA. This reaction occurs three times during each complete turn of the cycle. Thus, 6 molecules of PGA are produced. Reduction. The 6 molecules of PGA are first phosphorylated with ATP by the enzyme PGA kinase, yielding 6 molecules of 1,3-diphosphoglycerate DPGA. These molecules are subsequently reduced with NADPH and the enzyme glyceraldehyde 3-phosphate dehydrogenase to give 6 molecules of gal3p. These reactions are the reverse of two-step of the fourth process glycolysis in cellular respiration. Isomerization, condensation, or dismutation. For each complete Calvin-Boston cycle, one of the gal3p molecule with its 3 carbon atom is the net product and may be transferred out of the chloroplast or converted to starch inside the chloroplast. For the cycle to regenerate, the other 5 gal3p molecules must be converted to 3 molecules of 5 carbon RUBP. The conversion of gal3p to RUBP begins with a complex series of enzymatically regulated reactions that lead to the synthesis of 5 carbon compound ribulose 5-phosphate RU5P. Phosphorylation. The three molecules of RU5P are converted to the carboxylation substrate RUBP by the enzyme phosphoribullokinase using ATP. This reaction shown below completes the cycle. 3RU5P plus 3 ATP gives rise to 3 RUBP plus 3 ADP. Regulation of the cycle. Photosynthesis cannot occur at night, but the respiration process of glycolysis, which uses some of the same reactions as the Kelvin-Benson cycle, except in the reverse does take place. Thus, some steps in this cycle would be wasteful if allowed to occur in the dark because they would counteract the reaction of glycolysis. For this reason, some enzymes of the Calvin-Benson cycle are turned off in the dark. Even in the presence of light, changes in psychologically conditions frequently necessitate adjustments in the relative rates of reaction of the Kelvin-Benson cycle so that enzyme for some reaction changes in their catalytic activities. This alteration in enzyme activities typically are brought about by changes in levels of such chloroplast components as reduced ferredoxin acids and soluble components, example Pi and magnesium ions. Thank you so much. If you like the video, please like, share, and subscribe to our channel. To understand this topic better, please go and watch our previously uploaded video of photosynthesis. Thank you.